Welcome to AI for Good, the leading action-oriented global and inclusive United Nations platform on AI. Organized by ITU in partnership with 40 UN sister organizations and co-convened with Switzerland. The goal of AI for Good is to identify practical applications of AI to advance the United Nations Sustainable Development Goals and scale those solutions for global impact. In today's session, we're counting on you to use the live video wall feature to ask questions and post comments to help create an engaging discussion. We encourage you to stay until the end, as we will hold a 30-minute networking session in the neural network. Here you can meet, ask questions to our distinguished speakers, connect, and chat with the AI for Good community. It is now time to kick off the session and welcome our first speaker. The floor is yours.
All right, good morning, good afternoon, good evening, everyone, and welcome to today's webinar, which is co-organized by the ITU WHO Focus Group on AI for Health and the Department of Biomedical Informatics at Harvard Medical School. My name is Matthias Gröschel. I'm a physician and researcher working with AI for Good and helping putting together these AI and health webinars. If you're interested in learning more about this focus group or AI for Good, please don't hesitate to reach out. We're always looking for people to join the effort. I have the privilege of introducing the moderator of today's session, Anna Green. Anna Green is a computational biologist who loves to think about microbial evolution and informing computational models with biological data. After completing her undergraduate degree in molecular biology at the University of Connecticut, Anna did her PhD at Harvard University, where she developed new methods to predict protein interactions based on coevolution. Anna is currently a postdoctoral fellow at Harvard Medical School, where she develops novel computational methods to predict antibiotic resistance and infer the effect of selection on Mycobacterium tuberculosis genomes. Most of all, Anna is a really wonderful colleague. And with this, I'll hand over the word to you, Anna.
Thank you so much, Matthias, for that lovely introduction. And thank you everyone for attending the session today. I'm very excited to introduce today's speaker, Colby Ford. Colby T. Ford, PhD, is a cloud genomics and AI architect who focuses on the creation of scalable architectures to support computationally intensive tasks in machine learning and bioinformatics. He currently splits his time between various interests, including infectious diseases such as Plasmodium and SARS-CoV-2, neurology such as Alzheimer's disease and related dementias, and oncology using AI techniques to help develop potential diagnostics, vaccines, and therapeutics. And with that, I'll turn the floor over to our speaker, Colby. Thank you so much.
Thank you so much. Let me share my screen here. All right. Can you see my screen? We're good. All right. So today, my talk will be on scalable AI for good, specifically in genomics and health. So not only can we figure out how to gain predictive insights from genomics and push that over into the healthcare field, but how do we do this at scale? How do we do this at a large enough volume to have an impact on our everyday lives? Just a little disclaimer, this content in this presentation is public and can be reused with attribution, but nothing I say in this should be assumed to represent the opinions of my employers. These are my personal opinions. Just a tiny bit about me. I split my time between lots of different efforts. Currently, I run a consulting firm named Tuple, which is a cloud Microsoft partner and Databricks partner consulting firm focused on genomics and AI solutions. I also work at Seattle Genetics and AMISA, where I work on cloud architectures for oncology and Alzheimer's disease, respectively. And then I also teach in the School of Data Science at the University of North Carolina at Charlotte, where I teach cloud computing. My research interests kind of split between cloud computing and machine learning, but mainly using those technical techniques for sequence analysis and protein structure interaction. So the theme for my talk today is more about taking sequence information that we can get at a relatively large scale and using cloud-based scalable machine learning and computational services to then gain insights in things like diagnostics, vaccines, treatments, and then use those insights in public health. So specifically in this talk, I'll be talking about how to apply these techniques to things in malaria and COVID-19. which especially for COVID-19 is very relevant to all of us at the moment. So there are a few advancements that make some of this possible, right? One is protein structure prediction and docking. Obviously, we've all heard of AlphaFold. This kind of hit the news big last year. The ability to predict structures and not have to wait on experimental results to come out is super important in this. This is now done at a highly accurate level using AlphaFold 2 and RosettaFold, but then also understanding how proteins interact is super important for understanding their effects in the human body or in the body of other organisms. So we use tools like Haddock or LightDock, or there's a series of other docking systems that we use. But these tools individually are good at a single experiment, right? I can put in a single sequence or pair of structures and get a docking prediction out of it. But how do we scale that up into a higher level for higher throughput? So the next set of advancements that make this possible are cloud scalability tools, such as Apache Spark, being able to embarrassingly parallelize our tasks in the cloud, Docker to containerize these applications, which usually are pretty complex and have lots of bells and whistles that take a lot of dependencies to run, and then also scaling that up to dynamically run on GPU-based architecture, such as NVIDIA CUDA architecture, and then things like Kubernetes to run these things at scale, but also kind of clean up the compute environment once we're done. And then lastly, on the machine learning side, most of the times we want to do machine learning to make predictions, but I'm a firm believer that not only do we need to make predictions, but we could also use those models to then explain why something is happening to gain actionable insights into whatever it is that we're looking at. So the ability to automate and explain AI at a higher scale is super important for making all of what I'm getting ready to talk about possible. So for the first little section of the presentation, I'm going to talk about updating malaria vaccines and diagnostics using sequence and structural analysis. Part of this work has been published in Frontiers in Tropical Diseases, and then another is in review at the moment. So the first part is, when we look at diagnostic tests, and even if you've never taken a malaria diagnostic test, I think we've all probably taken a COVID test that looks similar to this. One of the issues is when these tests are designed, they use a specific molecular target as the thing to detect, right? So for a lot of malaria diagnostic tests, rapid diagnostic tests like these, they are looking for the detection of specific motif repeats. So the repeat of multiple patterns of amino acids in succession. Namely, some of the most common are using what are called type 2 and type 7 repeats that you see on the screen here, this AHH kind of pattern. These are called histidine-rich proteins, and specifically looking at histidine-rich protein 2. The issue, though, is that the reason why these two types are chosen as the motif is because of some modeling that was done back in 2005. Well, as we know, anything like from plasmodium to viruses to bacteria mutates as time goes on. And so now the accuracy of these RDTs has been decreasing due to mutations in this histidine-rich protein. So how do we pick better motifs for the RDTs? One option would be that we can start a bunch of lab-based experiments where we design different lateral flow immunoassays or the kind of primitive versions of those that we do in the lab, and then see, test this hundreds of times to see which of these motifs are the best things to look for. But this takes time, this takes money, and it just takes a lot of person power to do this. So what we've devised is a way of using machine learning to then predict which of these motifs should be used for future malaria diagnostic tests. So our investigation here is we took in 102 DNA sequences, and we counted the motif matches per sample. So these motif matches, for those interested, are called Baker repeats. There are 24 of them. There are a series of others that people have derived, but there are 24 basic ones. And these motifs are similar to what I showed on the previous slide. They all have a lot of histidine residues, and they all exist in the Plasmodium falciparum histidine-rich protein exon 2 and exon 3. Once we count the motif matches per sample, then we use a machine learning model to predict the infection. So across all of our samples here, we do know their level of parasitemia. We also know if they are positive or negative for Plasmodium or for malaria, given the PCR test as well as existing RDTs. So we kind of have these 3 levels of information. And then once we train this machine learning model and we're satisfied with the level of performance of the model, we can then use model explainability to then derive out which of the features are the most important across these 24 types. And the reason we do this is we can't create a lateral flow test that looks at all 24 types. And so we need to kind of cherry pick which of those are the most important across our data set, which what we found are types 3, 5, and 10 that give us the best performance. What's really interesting here is when we look at kind of the model comparison of the machine learning analysis, is that if we start with the types 2 and 7 that we originally started with, we get kind of mediocre performance, especially looking at when we're just looking at true positives and false positives from a ROC curve. Then if we put in all of the types in the machine learning model, which is not what we want to do for the test, but if we do this in the machine learning analysis, you see we get a little bit more consistent performance, but still a bit more mediocre. And then if we use the machine learning explainability to derive out which of those motif types are the most important, and then just feed those back into the machine learning model, you can see that we get much better and much more consistent performance across the board in our malaria prediction, diagnosis prediction. And so you see that, we really haven't changed the methodology at all of what we're putting into the rapid diagnostic tests. All we're trying to look for is how do we need to update these tests using the mutations or variations that we see in the Plasmodium population today. So since we can't test for everything in an RDT, which of the motifs gives us the best diagnostic power? And what we've determined is it's types 3, 5, and 10. And this is just an example of additional features that we could use in the future as well. So other RDTs are based off of, and even in other infectious diseases, are based off of all sorts of different types of molecular targets, from the presence of specific mutations or the presence of these motifs like this. And we can use machine learning to kind of derive out which things may be important. Continuing on from the diagnostic test issues is that we also see some issues with vaccine efficacy in certain parts of Africa, sub-Saharan Africa specifically. In Ghana, the RTSS vaccine has fairly low efficacy. It's about 30%. It varies between children and adults, but overall about 30%. When the RTSS vaccine was designed, it was designed based off of the Plasmodium falciparum 3D7 reference isolate, which is great, right? That means that if we see 3D7 existing in our population, and that's what people are being infected with, then the vaccine should be more effective. However, once we looked at our Ghanaian samples, we noticed none of them match 3D7 at all. And so what's going on with this vaccine, in the sense that if we wanted to update the vaccine for a specific region of Africa, is there a high throughput way that we can then try and kind of bubble up the specific haplotypes that we see in the population? So what the sequence that you see here on the screen is the Plasmodium falciparum circumsporozoite surface protein, PFCSP, and we're looking at 2 specific regions of that gene or that protein. So the TH2R and TH3R regions of that protein are the regions that actually vary the most as compared to the 3D7 reference, which you see in the top line here. What this means is these are the targets for CD4 plus T cells as well as CD8 plus T cells respectively. And you see what's happening here is that there is this, the Plasmodium is starting to mutate in those regions to potentially evade our immune cells. And so because of this, we then need to develop a better vaccine for the specific isolates or variants that we see in this region. So what we've taken to here is some protein structural modeling, which I'm quite excited about. Our first part of the investigation is to find SNP-based haplotypes. This is using a new R package called haplotype R. This is a way of finding, of all the variations that we see in our genetic data, which of those occur most often, and can we kind of cluster those up so that we're not looking at thousands of potential varied sequences, but a more manageable set of potential haplotypes. We then kind of filter and align these haplotypes to see how they vary from the reference, for the 3D7 reference specifically. And then we take to some AI-based computational techniques to simulate the degradation of that whole peptide or the whole protein into a smaller peptide. Going back a slide, this is for Th2R, we simulate the lysosomal degradation that actually happens in CD4 plus T cells. And then for T3R, we simulate that proteasomal degradation. We use tools like NetChop and NetMHC to then generate these peptides. And then we use a Python tool called Peptide Builder, which takes a sequence information and actually builds a PDB-based structure of that peptide, which is a small between 4 and say 10 or so amino acids long. And then we take that peptide and stick it in between the human leukocyte antigen and the T cell receptor, which you see here on the slide. So the T cell receptor being in green and the human leukocyte antigen being in blue. We take the CSP peptide, stick it in between, and then what we do is we use a docking system called Haddock to then see how the binding affinity changes depending on which haplotype's peptide we stick in there. And you may be wondering, why would we want to know this? Well, if we're going to choose a particular haplotype to update the malaria vaccine, what we would need to select a haplotype that commonly exists in the population in Ghana, but then also produces a peptide after degradation that induces a high enough binding affinity to be effective in the body. So once we do this process, we end up with a series of Th2R-based peptides or Th2R-based haplotypes, and then therefore these small peptides. And I won't go through each of these on the slide, don't worry, but just notice that across the board, there's a lot of them that have very similar binding affinities. And then sometimes you see a big shift in the docking position, not only of the T cell receptor to the human leukocyte antigen, but also just the positioning of the peptide within the groove of HLA. You can see longer peptides, like for example, in CSP22, sometimes bulge out of the groove in HLA. And then some of them have different positions altogether. And so we really wanted to look at, the presence of particular residues in a peptide that results in from this analysis. Does that cause some major thing? Like, do we need to avoid a peptide that includes tyrosine? Do we need to avoid a peptide that includes other residues that could create other types of bonds or other types of polarity in the molecule? And we didn't see any huge difference there, but we did see differences across the binding affinity here. And once we see which of these haplotype or which of these candidate peptides have the best binding affinity, then we can take those kind of top contenders and see which ones actually exist most commonly across the population in this area, in Ghana specifically. Similarly, we looked at the T3R region and noticed that there really are only just a few haplotypes that pop out of this analysis, meaning that despite there being a considerable amount of haplotypes in general across the population, they all produce very similar peptides. So after degradation, the peptides are very similar, and they all have, kind of spoiler alert, if you want to read the paper, they also have very similar binding affinities there. So perhaps the T3R region for CD8 positive T cells is not really the region that we need to focus on as much. It's really the massive variability that we see now in CD4 positive T cells with the Th2R region. So what we see with this is that what was really interesting is that when we look at the peptide and how it binds in the HLA groove, we see that the larger the peptide, the better. And what you see here is a Haddock score, and the lower the Haddock score, the better binding. And so, for example, the longer of that peptide sits in that groove, the more context it can have, the better that binding is. But oftentimes, the better that binding is, the more it bulges out of that groove, and then it reduces the binding affinity of the T cell receptor. And so if you know the story of Goldilocks, we almost want to take this approach where we want to find something that's not too small of a peptide, because then it doesn't have a high enough binding in HLA, but we don't want to get a peptide that is too large, or else then the T cell receptor doesn't bind well at all. And so what we want to look for are things that are somewhere in the middle. So for example, if I take the haplotype, which is called CSP35, and it's also the same resulting peptide for CSP39, which is DIE-LIQN. This is found in 29% of our samples, so a pretty large chunk of our sample set here. This could be a good contender for updating the malaria vaccine in this region, because it not only exists at a high enough proportion in the population, but also after degradation in the human body and expression via our antigen presenting cells, it actually works quite well and docks at a sufficient enough binding affinity between our human leukocyte antigen and T cell receptor, at least in our simulations. All right. For the second half of the talk, I want to talk about some of my SARS-CoV-2 work, which I'm also quite excited about. This was just published, I guess, early last week in Frontiers in Virology. And this goes into the structural analysis of the antibody binding of potential SARS-CoV-2 variants, specifically the Omicron variant that came out in November. So the problem that we see here is that every time we turn around, there's a new variant popping up, variants, and more variants. And what ends up happening is that, as these variants pop up, it poses the question, or posits the question of, okay, is this variant serious? Is this something that we should care about from a public health perspective? Is this something that we should care about from a vaccine perspective? So if we've been vaccinated already, does this mean that we need to go get another vaccine or maybe a booster? And even if we have been vaccinated, what does this mean from a transmissibility and pathogenicity standpoint? So it posits a lot of public health questions. And so while we're waiting on the actual experimental results to come back on the actual structural determination of any SARS-CoV-2 variants, can we predict if a variant is serious using computational techniques? And spoiler alert, yes, we can. So how this work started, it was quite interesting. I was actually sitting on the couch right before Thanksgiving, and I was getting questions by family members about Omicron and how is this actually serious? And so I didn't know the answer to the question, so I took to these computational techniques to try and derive out some predictive answers here. So once we get the sequence information, which actually comes out quite often or quite quickly for new variants, and these are posted online, we can take these input sequences and then predict the structure of the region of a particular protein that we're interested in. So for example, once the Omicron sequence came out on November 22nd, I could then take the region of that genome that I care about, which was specifically the receptor binding domain of the spike protein, and I can take that and predict the structure using one of multiple tools, for example, AlphaFold 2 or Rosetta Fold from the University of Washington. Once we have those predicted structures, I can then compare that to a reference structure just to see if the predicted structure is believable or not. Because sometimes with these predictions, you get some predicted structure that looks way off, and we know that we don't need to proceed with that. Once we get this predicted structure, I can then bind or dock that this predicted structure with a target structure. Here, the target structure being one of multiple different neutralizing antibodies that were available in Protein Data Bank. So all publicly available data, and I can quickly do this in the matter of a few hours. So once I take the target structure or the antibodies and stick them on top of the receptor binding domain, I can then get predicted docking metrics out of this to see how that binding affinity is changing. And then I can also get the predicted docking structures, the proteins in complex. So to begin with, once we have the RBD structure or RBD sequence, excuse me, from B11529 or the Omicron variant, I can then push these through AlphaFold and Rosetta Fold to get a structure. What's really interesting is, so I performed this part of the analysis on November 22nd or 23rd, but the actual Omicron actual structure, PDB17-9J, was actually not released until 12/31, so the last day of the year. So over a month ahead, we were able to get a structure that is actually quite accurate, especially the AlphaFold one that had an RMSD when comparing the orange structure on the screen, the actual structure, and the blue structure of less than an angstrom. So very, very accurate, especially for the backbone structure, just the backbone conformation. And then once we use once we find particular neutralizing antibodies that are available on Protein Data Bank, these are publicly available, we can stick these on top of the predicted Omicron structure or the actual Omicron structure, which I did later on, or as a baseline reference, we need to understand what's the predicted docking between the antibody and a previous isolate or a previous variant that we know. So for example, alpha. So if we take the actual alpha structure and stick the antibody on top of it, we can get kind of a baseline reading of that docking affinity. And then we can then compare that kind of baseline reading against the Omicron structure, either the one that came out of AlphaFold 2 or Rosetta Fold in blue and purple. And then later on, once the actual Omicron structure came out, then we can kind of sort of validate these results of now that we know what the actual structure is, what does this mean? And you may be wondering, what does this actually tell us? This tells us that if someone has been infected with alpha already, or has had a vaccine with the alpha-based mRNA vaccine, then someone could potentially be reinfected, or you could have a breakthrough infection. But the binding affinity is high enough with these neutralizing antibodies that we've tested here that we see sufficient enough activity. So meaning that for any of us that have had Omicron, which I have, and after being vaccinated 3 times, the infection was very mild, if anything, which is really interesting to know from a public health perspective. Just a note here, this is not the full antibody structure that you see on the screen. It's only the heavy and light chains of a single fragment antigen binding region. So what we see from a docking metrics perspective is that we can, given that we have multiple different neutralizing antibodies, we can kind of see how this changes between the reference structure, the alpha structure that we have, and then across our predicted structures, and then our actual Omicron structure. And what you can see here across the board is that the AlphaFold prediction in this case is more closely related to the actual Omicron structure that came out later on. But we can then see how do things change? Like, for example, van der Waals energy or electrostatic energy, that interaction between the two, the antibody and the receptor binding domain, how does that actually change when we look at alpha versus Omicron? And if we have a big drop in binding affinity, if we were to have had a bigger drop in binding affinity, then we could have posited that there would have been a bigger public health effect. Outside of malaria and SARS-CoV-2, just in a general theme of the structural work is that there are huge therapeutic design opportunities. One is that we could have a single drug, and we want to test that single drug against multiple candidate target proteins. And so if you work in therapeutic design in monoclonal antibodies, this would be where we want to stick a particular antibody, a candidate antibody or drug against multiple targets. Or maybe we have a single target that's of interest, but we have multiple drug candidates. We could test that as well. Oftentimes when we design therapeutics, we know that something may be active in the body or seeming to bind and be active against a particular target, but we don't actually know where it binds, and therefore we're not really sure about its mechanism. And so these structural and docking analyses that I presented here could be used for epitope or binding site prediction. And not only that, but let's say we are a drug company and I'm creating a therapeutic that seems to be really, that seems to work really well, but then for certain people in the population just doesn't work as well. And is that because of a particular variant or genotype that exists in the population that causes the particular drug not to bind as well due to a mutation on that target protein? And so we can test all of these things in silico without really having to wait on lab results. And just in summary and in closing here, these deep learning based analyses really allow us to make educated guesses very quickly. So there's obviously a big need to validate any of these results, especially in drug design or vaccine design, for example. But we don't necessarily have to wait for all of those results to come in to sort of gain confidence or gain some insights into the questions that we have. For example, the SARS-CoV-2 stuff being finished a month ahead of time there. We can now rapidly generate structures that are quite accurate and then dock them to therapeutics months before the experimental results are available. So like what you see here on the screen is an antibody bound to the receptor binding domain. And then we can employ machine learning model explainability, not only to understand the prediction and to get good predictions out of these systems, but understanding the how, answering the questions of how would we implement this kind of computational prediction into something that's actionable in the real world, for example, in vaccine design or the design of diagnostic tests, as I've shown in my malaria work. And future capabilities are to scale up these analyses into something where we can really perform a lot of these analyses at scale very quickly. And that's something that I'm working on, for example, in the ability to use AlphaFold 2 inside of Azure Machine Learning, a cloud-based machine learning product, to where now not only do I need to just run a single sequence, but I can run a series of tens, hundreds, thousands of sequences in a cluster and get the results in about the same amount of time. So given enough compute power that you throw at it, we can get our results very quickly. And then the next step is to enhance our docking capabilities because the docking analyses here are quite slow in comparison to the structural prediction. We want to kind of scale that up and make that faster and more reliable, especially when we know what the binding site is. So for example, if we just wanted to do a comparative analysis from a series of therapeutics to a single target, how would we do that if we know what that target is and we would just want to get some quick results out to understand that binding affinity change? And that's the next thing that I'll be working on. And with that, I can now start taking questions.
Great. Thank you so much, Colby, for the talk. I'd just like to encourage all the audience members to go ahead and ask questions on the Neural Network website. If you post your questions in the chat there, we will see them and I'll be able to forward them to our speaker. But in the meantime, since I'll just take moderator privilege, I guess, and go ahead and ask my own questions. So first of all, I thought it was very interesting how you showed how we can use machine learning and computational techniques to kind of update what we think our therapeutics to malaria should be based on changes that we've seen in the virus. And I was curious, like, do you think these technologies provide the capability to move from kind of a reactionary stance to a predictive stance? Like, can we predict which epitopes might be the best for creating a vaccine that would be hard to mutate away from, for example?
Yeah, absolutely. I think there'll be a bigger need as we see things like mRNA vaccines being easily created, as in we can more rapidly create it than traditional vaccines. I think what we'll see is that we may even have regionally specific or country specific vaccines for various diseases, whether that's SARS-CoV-2 or malaria or anything, HIV or anything else, right? It's where we can actually target the natural variation of that organism in an area. And so I think not only that can we get really specific with our vaccine design, but we can also get specific with the location as well. We know that within SARS-CoV-2, for example, the virus mutates in areas where there's selective pressure. So when we have a vaccine or antibodies that are binding to a specific region of the receptor binding domain, then we see a lot more mutations in that area. But could we develop a vaccine or antibodies that bind to a different site that have other inhibitory processes or mechanisms? Potentially, absolutely. So for example, maybe we don't actually have to block the top of the receptor binding domain, but we could block another part of the spike protein so that it can't even enter our lung cells, for example. So that's one option that we could certainly do with these techniques.
Yeah, absolutely. Or even identifying certain residues in the spike protein receptor binding domain that are critical, that are hard to mutate. Yeah.
And we can also simulate these things too. So before they happen. So if I wanted to, some work that's not shown here today, but if I wanted to just mutate a sequence, go in with my text editor and change a residue, I could then predict what that effect, what that mutation's effect would be on the overall structure, and then potentially binding with current antibodies.
Mhm. Yeah, I think that's an awesome possibility. Matthias, I don't know if you have any questions you would love to throw out there.
Colby, yeah, thanks so much. So do you have any plans to, or have you worked and talked with, folks at CDC of kind of integrating your predictions? I mean, because they're certainly going to be busy at looking at all new variants and trying to identify if they will be variants of concern or not. Have you talked to them?
Not specifically with SARS-CoV-2, but we have talked about kind of what's the next pandemic. So after SARS-CoV-2 or COVID-19 kind of becomes a background infection like the flu, what's the next thing that could pop up? And so we're looking at things, for example, in flaviviruses, so dengue, yellow fever, things like that, where, if those were to become an actual pandemic, it could be quite serious as well. So we're kind of being a bit more forward looking at that to see potentially what therapeutics could be reused. Also, if what variants exist in those populations today that could cause a problem in the future. So to answer your question, yes, just not exactly on COVID at the moment.
Do you know how currently variants of concerns are being defined? Do they use modeling? Do they just align the epitopes?
Yeah, so variants of concern, there's multiple ways and there's multiple groups. Variants of concern really are defined by the World Health Organization, really from a global perspective, but then more locally, by the CDC and equivalent agencies. The initial way that this happens is that once a variant is determined to exist in the population, we then need to see what's the prevalence of that variant. Is this something that's just like a one-off mutation that one person had, or is this something that a group of 50 people have, and then all of a sudden overnight it turns into thousands, which is what happened with Omicron. And then there's a lot of phylogenetics that go on. So there's a group called Pangolin that developed the lineages and named the lineages, and then based off of how different those lineages are from other variants, then we can call it a variant, give it a name, which is quite confusing, even for someone like myself who works in this area. It's quite confusing to understand the naming conventions of this, but yeah, the variant of concern is it's multiple facets. It's how different is it, how prevalent is it, and then also how serious is it, as we see, because then things get elevated into that concern status based off of those conditions.
Yeah, absolutely. I think for outsiders, the variant nomenclature can be quite confusing too. I think
even for insiders.
Learning that it's a variant is like a pool, like a population in which there is genetic diversity. So like in our terms in TB, it would probably be more like a lineage or something.
Yes.
Yeah. Since we're still waiting on some audience questions, I guess I'll continue to pick your brain if that's all right.
Sure.
I was curious, kind of what kind of experimental data, if any, you need to generate a mapping between docking affinity and Haddock and like in vivo capabilities, right? So what's a Haddock score that means something's not going to function in nature?
Right. So the Haddock scores functionally, and there's different scoring across the different docking systems. A Haddock is just what I used here, but Haddock score is a kind of conglomerate score across electrostatic energy, van der Waals energy, and I think desolvation energy. So it's kind of just an aggregate score of those three, a weighted aggregate score. The number itself doesn't really mean much, because if I were to dock two different proteins, you might get a score of -80, which is good. But then if I docked two other things, I might get a score of -112, and that may seem better, but it's all very relative. And so that's why you need to use things like what I did in this study, which is dock to alpha, which is known, like the alpha variant of SARS-CoV-2, and then you get a baseline, and then you can compare from the baseline. That being said, we do know that there are kind of correlations between certain things like van der Waals energy and some binding affinity methods that exist in the lab. And so I'm still working to kind of validate those and see which of the experimental metrics are kind of proxies for these metrics that we get out of the system. That being said, there are ways of deriving out things like KD, for example, the binding affinity with that, and then also there's other methods to actually determine like number of contact sites and things like that as well.
Yeah, that's very useful too, that you can use known structures with known affinities as baselines. Because the concern with me, like I have used Haddock before as well, it's always like, how exactly does this map to in vivo behavior? But if you have something with known behavior, that's very useful. Yeah.
To quote Einstein, it's all relative, right?
Yeah, yeah, absolutely. So I think perhaps it might be prudent for us to go ahead and move over to the neural network a little bit early so that we can interact more with the audience, if that sounds good and there are no objections.
It works.
Okay, great. So I will see you guys on that platform shortly. Thank you so much to everyone for attending.